and it is brutal. And to be successful requires incorporation of the fact that it's a meritocracy on a global level, and there is only one standard, which is the absolute best. So my frame of mind when I grew up and was educated and was prepared to start my own laboratory as a young professor and to take on Professor Philosophy, all from this very fundamental assumption that every process that occurs in a living cell, however complex, even the very processes that generate the organization of the cell itself in 3 dimensions within the cell, even these processes can be reproduced in a cell-free extract, can be treated as chemical processes. And so in the early 1980s, my colleagues and I set out to see, with this idea, could we take this philosophy of Edward Buchner one step further and reproduce the most complex process to have ever been studied in a cell-free system, the organization of the cell in 3 dimensions within itself. And to the great surprise of many people, and I must admit even myself, it turned out that this was possible. It took a lot of work, and I'd like to acknowledge, and it's something that is very important, and those of you who are students, I hope will appreciate, there is a lot of hard work that goes into doing this scientific research. And it does not happen only from the professor. Though in those days I was very young. I was 29 years old. I am now 62 when this was first done. Now you see why the phone call was considered long overdue. Okay? My colleague, Eric Fries, who is now a professor at the University of Uppsala in Sweden. Eric Fries was 28 years old at the time. And together, we ventured into the laboratory with a homogenizer. This is the tool that is used to pulverize the cells. Not very different than the device used by Edward Buchner in 1900, but used for a different purpose. In fact, it was this very homogenizer that was used in the first reconstitution of cargo delivery in a cell-free extract. And it now resides, thanks to Eric Fries, who saved it after all these years, in the Nobel Museum. We were asked to make a donation to the Nobel Museum in Stockholm. And so it sits there as a memorial to this work. And I won't go through the technical work that was accomplished here, but briefly, we take cells from the body that are cultured. They're human cells, but they are grown in culture for many years. And we disrupt them with this homogenizer. And we find ways in the test tube to incubate these as if it were a chemical reaction. And we add back the sap of the cell, which is called the cytosol. We add back the source of metabolic energy in the cell to power the trafficking process, just like gasoline is needed to power a Federal Express truck. ATP is needed to power most every process in the cell. And we have developed a method to measure the transport of the cargo. Again, this has been published for many years. It's actually in many high school textbooks, so I won't give you the details. But the basic idea is that when the cargo gets delivered, it is modified chemically. And we can add a radioactive. And they said, no, you're wrong for this reason. You're wrong for that reason. You're wrong for the next reason. Well, they may well have been right, but they weren't. And it turns out that this is a very important process of the scientific discovery process. You make a step in a fog, and you see a little bit of light. You interpret it a certain way. You maybe think you're right, but you don't absolutely know. And so you have to test it, and you need to be challenged. And this process of challenge takes 10 years or more. And eventually the result is that the entire establishment really begins to accept the truth of a proposition that otherwise seemed too simple to be true. And what was the idea? The idea that came out of this is that every vesicle, every delivery truck, having cargo within it, is able to find its matching compartment destination by way of partnering snare proteins. Do you remember I mentioned the idea of Velcro? Imagine that there is a green Velcro and a red Velcro. And this Velcro, for those of you who have a background in biochemistry, takes the form of an alpha helix of protein. It's a very basic structural motif in protein, and alpha helices have the ability of gravity. There must be universal gravity. And then he says, I can explain how the moon circles around the Earth because this apple fell. That is crazy. I mean, anybody that would say that is crazy. But of course, he invented modern physics by saying that. He believed that there is a universal law that can explain everything, one law. The physicist believes that. So I have approached biology with the idea that it must be simple. And if you try hard enough, you will be able to formulate a simple explanation. Now, the reason that was important in solving the problem that we solved, and the problem that we solved was, how does each of these little vesicles know to go to the right place to deliver its carbon? Because if it went to the wrong place, the cell would lose its organization, and it would never grow, never develop, and we would die. Okay? That's how fundamental this is. How then does each delivery truck, each vesicle, know to go to the right place? At the time that I took this problem up, in about 1980, my colleagues said, well, it must all have to do with the anatomy of the cell. For example, this membrane-bound system here is called the endoplasmic reticulum. It is where the proteins, the cargo, are first made. The next place that these proteins go is a packaging station called the Golgi apparatus, named after Professor Golgi, who received the Nobel Prize in Medicine in 1905. That's how old this process is. Now, how does this vesicle, this cargo, this FedEx truck, carrying the cargo, know to go from here to there and not to someplace else? How does it know? Well, the wisdom in the field was, it's very simple. Here is the origin of the truck, the endoplasmic reticulum. There is the destination, the Golgi apparatus. Look how close they are in the cells. It's very simple. These trucks are leaving here, and they have no place else to go. They are being fed directly into this destination. According to this, it's the anatomy that generates the specificity. The practitioners in this field, the scientists, very educated people, were microscopists. They were anatomists. Professor Pilati himself was trained in anatomy in Romania. He was an anatomist. So for them, it all had to do with how things are arranged. I came from physics and chemistry, and I thought that's much too complicated, because how could you generate that arrangement in the first place? So I approached it from a different point of view. My idea was that each of these vesicles, each of these delivery systems, must have on its surface a specific chemical, a receptor, that says, I am destined to go here, not there. Intrinsic chemical specificity. And there must be, at each membrane to receive the cargo, another molecule, a receptor, and the two receptors bind each other in a very specific way, so as this vesicle can always go there. It doesn't have to be fed into there. It could go off here, diffuse away, and then come back. You will always end up at the right place, because like Velcro, the vesicle has Velcro that sticks to the target. And if you imagine in the cell that there are 20 different types of Velcro, each complementary to the other, then the cargo can always be delivered at the right place. That was my idea, intrinsic chemical specificity. And the importance here is the specificity dictates the anatomy. In other words, it's all chemistry. And if you started out without this anatomy, in a hypothetical cell, if you went in and stirred up everything and rearranged it all, and it's random, it would automatically come back according to chemistry, and it would automatically resume its organization. The anatomy follows from the chemistry. That's a radical idea. Now, that idea, though it may be radical,